Hi, everyone. This video is to go along with those interactive slides on Gregor Mendel and an introduction to genetics. So Gregor Mendel is a really interesting individual, and he is what really piloted and pioneered this whole concept of genetics, which is so important and valuable today in so many different fields of science. So I'm going to talk you through this, and we're actually going to go through those interactive slides together as you fill it out. So let's start off by beginning, what is the difference between genetics and heredity? These are two terms that are oftentimes used, but not always specifically defined. So heredity is the passing of traits from parents to offspring. However, genetics is the study of how those traits are passed on from parents to offspring. Even before Gregor Mendel existed, it was clear that oftentimes kids looked like their moms and their dads, but genetics is what actually helped explain how those traits were passed on from parents to offspring. So let's get started. Who is Gregor Mendel, and what the heck does he have to do with genetics? Okay. For a very long time, people understood that traits were passed down through families. However, how this occurred was really unclear. Gregor Mendel's work with pea plants, like peas, those little green things that a lot of you guys don't like to eat, were crucial to help people understanding how these traits were passed down through generations. So a little bit about Gregor Mendel. He is considered the father of genetics. He was the very first person to track one trait through several generations. He looked at different types of traits and saw how they passed down through generations, much like I can see how my butt chin was passed down through several generations, okay? In the late 1850s, so think about how long ago that was, he grew and studied pea plants. He was an Austrian monk and a biologist, and his work was not really recognized until the turn of the 20th century. So the very first question is, drag the circle to the correct response. Which of the following is true about Gregor Mendel? He was an American monk? Nope, he was an Austrian monk. He carried out research with Darwin? No, totally different times. But we also didn't mention that yet, so we know that's not true. He was a zoologist? Nope. In the previous slide, said he was a biologist and an Austrian monk, but he was considered and is still considered the father of genetics. So you're just going to, didn't mean to zoom in on that, guys. Sorry about that. Whoop, let's turn that off. He is considered the father of genetics, okay? So what are traits? They are distinguishing characteristics that include butt chins, or cleft chin is another name for it, okay? Eye color, nose shape, hair color, skin color, the fact that you might have ear hair. There's a lot of fun traits that some of you guys may want and some of you guys may not want, okay? So how do people get their traits from their parents? Well, traits are inherited from your parents. Half of the genetic material comes from your father and half of the genetic material comes from your mother. In the previous unit, when we were talking about cell division, mitosis and meiosis, we get 23 chromosomes from the egg cell or your mother and 23 chromosomes from the sperm cell or your father. And when the sperm and the egg meet, they don't just make an embryo that develops into a human, but they also combine those 23 chromosomes to make a total of 46 chromosomes for you. So Mendel decided to study how exactly are these traits inherited by pea plants. He went out to the garden where he lived, and he decided that pea plants would be pretty easy to study because they can be self-pollinated or cross-pollinated. Also, they don't take that long to produce. Think about it. If he was doing this study on humans, he'd have to wait 9 months for that baby to show up. And then you've got to do it all over again for one kid most of the time. That would take a long time. But pea plants, you have a lot of different peas, you have a lot of different plants, and you can reproduce them very, very quickly. So what is self-pollination and what is cross-pollination? Well, self-pollination is when you pollen from another plant is transferred to the stigma of the same flower. In cross-pollination, pollen from another plant is transferred to another flower. So self-pollination, you've got one flower, okay? You are taking the pollen from that flower and fertilizing it. it's pretty much reproducing by itself. It's not like with people where you need a sperm and an egg. It can actually be pollinated by itself, my hand being the flower, okay? Cross-pollination, you've got two flowers, okay? You're taking the pollen from this plant and bringing it to this plant, okay? This is more like what happens in the animal kingdom, okay? So, drag the words to the proper banks, okay? Self-pollination and cross-pollination. Well, pollen from another is transferred to the same stigma of the same flower. That would be self-fertilization, okay? Or self-pollination. If I can click this, there we go. Sorry, guys. The computer's being silly today. Okay, self-pollination. Pollen from another flower is transferred to another flower. So this would be two different flowers involved. That's going to be cross-pollination. All righty. So what characteristics did he study? When you look at a person, you can look at hair color, you can look at hair texture, you can look at whether or not it's curly or straight or wavy. You can look at, are they balding? What type of balding? Do they have male pattern balding? Do they just have balding spots? Do they not grow hair at all? Then you can look at skin color. There are a vast spectrum of skin colors. You can go into eye color. You can go into ear shape, whether or not you get ear hairs, whether or not you get nose hairs, your chin. There are so many different traits with people that we can look at. With peas, there are still a lot, but not quite as many to sift through. So he looked at seed color. Were they green or were they yellow? Seed shape, were they wrinkly like this one right here? Or were they smooth and round? Plant height, were they tall or were they short? Pod shape, were they round at the bottom or were they straight? The pod shape itself, the pod color itself. So not just the pea color, but is the pod that holds those peas green or yellow? And a lot of other things as well. So let's talk about one example of Mendel's experiments. So just like with reproduction with humans, we have parents, okay? A mother and a father, a sperm and an egg. Granted, we can have different combinations of parents, but biologically, we do need a sperm and an egg to reproduce, okay? So that is called the P generation or the parent generation, okay? That's where that term comes from. So the parent generation or the P generation is when true breeding plants, so plants that always produced identical offspring, okay? So let's say we had a plant that was tall with green peas. Every time it reproduced, it would keep reproducing tall plants with green peas, okay? So plants with purple flowers and plants with white flowers. Every time that plant with white flowers would reproduce, it would have white flower offspring. Every time that purple plant would reproduce, it would have purple offspring. That's the parent generation, okay? The OGs, the original generation. Now, when you cross those parent generations, so maybe you have one plant, I've actually got a direct example right here on the page, so I don't need to show you my hands. We've got one parent generation that is purple. Okay, and that signifies capital W, capital W. Purple is dominant. So this is homozygous. Homo means the same. Homozygous dominant, that capital letter that overshadows that recessive trait. So that's one true breeding plant. The other true breeding parent generation is going to be this white plant. It's little w, or homozygous recessive. Remember, in this case, big W would be purple. Big W would be purple, but it would be called heterozygous. And little w would be homozygous recessive, and it would be white, okay? When big W and little w cross, that's the parent generation. The offspring is the F1 generation. So here are the F1 generations. You see that all of those offspring are purple, even though it had a white parent. Now, I want you to think about this. You get 1/2 of your DNA from your mom or the sperm, and 1/2 of your DNA from your dad or the mom, egg, dad, sperm, okay? So each one of these is going to get a big W. But what is the only thing that this white one can give? A little w. And remember, if that dominant allele shows up, it's going to overshadow that recessive allele. So all of these are going to be big W, little w, but they're all going to be purple. This is why it's important to know the difference between a genotype and a phenotype. A genotype are those two allele combinations, big G, or big W, which is homozygous dominant. That's going to show the dominant phenotype. Big W, little w. Its genotype is heterozygous. It's going to also show the dominant phenotype. Little w, that is homozygous recessive, and that's going to show that recessive phenotype, or the white. None of these are little w, so none of the offspring are going to be white. Now, where did the white flowers go? He called the purple trait the dominant trait. Purple is dominant. When it's dominant, just like a dominant person, they sometimes are overbearing, right? They dominate the conversation. The other person, it's almost like they're not there, even though they are. This is a lot like how dominant and recessive traits work themselves. So plants are called hybrid if they come from true breeding plants, parent plants, with different forms of the same trait, okay? So the parent generation, or the P generation, big W, little w. Their offspring are hybrid or heterozygous. So this one is not going to be big W. It's not going to be little w. Instead, it's going to get one of those big W's from the purple plant and one of those little W's from the white plant, and it's going to be hybrid or heterozygous. Now, if we cross those hybrid crosses, okay, so the parent generations are the OGs, okay, white and purple, homozygous, recessive, and homozygous dominant, they cross, they give us all purple or heterozygous. That's going to be the F1 generation. Now, this would be weird in mammals. However, in plants, it's not weird. We're going to take those offspring, those F1 generations, and we're going to cross those, okay? So these are not big W. These are big W, little w. When they cross, their offspring has a 25% chance of having a white flower. We're going to go into that shortly, okay? So in the F2 generation, the white flower appeared. That white flower or that white trait had been in the genes of those purple flowers. However, it was hidden by that big W or that dominant trait. Mendel calls that white trait or that white allele recessive for that reason. So now we're going to drag the lines to match the correct generations and experiment summaries. Okay, so these are the OGs, okay? This is big W, and this is little w. What would those original plants be? The F1s, yes. I didn't do that right. There we go. F1s, okay? Oh, that's not right. The two true breeding plants offspring, that would be this one, okay? When we cross those two, this is what the offspring is going to be. What are these two? Forgive me. Those are going to be the parent generation, the original generation. And last but not least, the two hybrid plants self-fertilize and produce offspring that are 25% purple and 25% white. That would be this one right here, okay? So the parent generation is the very top. It's when you have big W, and little w, those homozygous, recessive, and dominant. That's the parent generation, the originals, okay? When that parent generation crosses, they're all going to be hybrid or heterozygous offspring. They're going to show that dominant phenotype or purple. That's going to be the F1. Now, when that F1 self-fertilizes, remember it's heterozygous, so it's a big W and a little w. We're going to do a Punnett Square in a moment to explain this. When that crosses, it's going to give us the F2 generation, which will then give us 75% dominant, 25% recessive. So Mendel conducted these same experiments with all traits when he studied and discovered the same pattern each and every time. Excuse me. Dominant and recessive traits. So a dominant trait is the recessive factor that blocks another factor. Just like a dominant person in a conversation blocks the recessive or the quiet person. A recessive trait is the genetic factor that is blocked by the presence of a dominant trait. This is like blue eyes, okay? I have blue eyes. My husband has blue eyes. Our child has blue eyes. Likely, not always, because eye color is a little bit unique, which we'll get into later on this unit. It's not just dominant and recessive. There's a little bit more to it. But if my husband had really dark brown eyes, likely our child would be a carrier or hybrid for blue eyes, but he wouldn't have blue eyes, okay? So what were Mendel's conclusions? Well, Mendel concluded two factors. One from each, the sperm, and one from the egg, control each inherited traits. We now call these allele. So if your dad or the sperm had big W, and the egg had big W, little w, you're going to get one big W from dad because that's the only thing he can give you. But mom, the egg, has big W and little w. So there's a chance she could give you a big W or she could give you a little w. Those are called alleles. So those individual letters are called alleles. So let's drag the line and match it with its definition. So we have trait, inherit, dominant, recessive, gene, and allele. Let's start with dominant. We know that dominant is like the loud person in the conversation. So let's find which definition goes along with that. A genetic factor that blocks another genetic factor. Yeah, that sounds a lot like it. Let's see if there's anything else. Section of a DNA that controls a trait, genetic factor that is blocked by the presence of a dominant factor. That sounds like the opposite of dominant. A distinguished characteristic representing different factors of a gene. Dominant is going to be a genetic factor. Keep opening up the dictionary. I really want to know my vocab. That blocks another genetic factor. Now, let's go ahead and do recessive because I heard recessive. Dominant is what overshadows it. Recessive is what is overshadowed by dominant. So that would be the genetic factor that is blocked by the presence of the dominant factor. All right, let's talk about inherit. Okay. When you inherit a trait from your parents, you are going to receive it genetically from your parents. So which one of these explains that? To receive a form or from a parent genetically. That would be inherit. All right. Got 3 more to go. Which one, trait, gene, or allele, would be, or a distinguishing characteristic would be trait? Section of a DNA that controls a trait? That doesn't make sense. It's got the vocab word in it. A distinguishing characteristic? Maybe. Represents different factors of that gene? I would say that a distinguishing characteristic is definitely that answer. All righty, now we've got gene and allele. These are commonly confused. Remember, allele is like a letter. Think A is the beginning of the alphabet. It's going to be a letter. You're either going to get big W or little w. You're either going to get big B or little b. So which one of these would explain that? Or different factors or measurements of that gene? So that would be right here. So that leaves us with gene, section of a DNA that controls a trait. All righty. So let's go over homozygous versus heterozygous. Remember, homo, let's talk about those prefixes, is the same. Hetero means different, okay? So if the alleles are the same, and alleles are those letters, they are considered homozygous. If alleles are not the same or are different, they're considered hybrid or carriers or heterozygous. So homo, the same, hetero, different. So here we have big W, and big W, little w. Those are both homozygous. This one, big W, would be homozygous dominant. Little w would be homozygous recessive. If the alleles are different, they are considered heterozygous. All right, so let's go through this together. Drag a circle around heterozygous genotypes and drag a square around homozygous genotypes. Remember, the genotypes are those combinations of allele. Phenotypes are those physical characteristics. So let's go ahead and do all of the homozygous first. Remember, homozygous is the same. So this would be homozygous. little n, little n. This would be homozygous. Big little h, little h. Oh my gosh, I'm doing the wrong ones, guys. Forgive me. Homozygous is going to be square. So little n, homozygous. Little h, homozygous. Little e, homozygous. Little p, homozygous. Big Y, homozygous. And then these are different because there's a tall and a short, or a dominant and a recessive allele. So those are going to be heterozygous. That's what we're supposed to drag those circles around. So big E, little e. Big L, little l. And if I can grab this one. Big U, little u. Okay? So genotypes and phenotypes. Genotypes are the possible combinations of those alleles. So the genes, big W, big W, big W, little w, little w. The phenotype is that physical characteristic. Big W is the genotype. The phenotype for that is purple. Little w is the genotype, but the phenotype is white. Big W, little w is the genotype. Big W, little w's phenotype, though, would be purple. So let's go ahead and sort those. Phenotypes. These are the physical characteristics. We've got round, big T, little t. Those are two alleles. That's the genotype. Big B, that's the genotype. Yellow, that's a physical characteristic. Little y, that's the genotype. This dictionary. And short, that's a physical characteristic. That's the phenotype. So in order to visualize how these traits could be distributed amongst the offspring, you can use a Punnett Square. We're going to have an Amoeba Sisters video that goes over this and really, really well. And we're also going to have office hours and time in class to go over this together. But essentially what's happening is, in order to determine what that phenotype is, we can actually plug the genotype of the parent. So let's say the egg has big A, little a. Big A, little a. And then let's say the sperm has big A, little a. Big A, little a. If you have 25% chance that the mother and the father are going to give both a big A and a big A to the child, you have a 50% chance that the child is going to get a big A and little a, or the offspring rather. And then you have a 25% chance that the offspring is going to get a little a and a little a. If you guys have questions, let me know. I hope this helped out. Thanks, everyone.